Okay. All right. So I'm recording. So we're here to group activity 5 or any other questions you have besides that. But I particularly wanted to get people to group activity 5. So we got 2 more next week, 6 and 7, that we're doing lab, but we got to get past 5. Okay. So have you guys done at least part of group activity 5 or started it or not?
What was the question?
Oh, well, have you done any of group activity 5 or are you just starting it or?
Yeah, I just started it today.
Okay, wait a minute. Wait, my phone is ringing. Hang on a second. Hello? 106. Okay, well, let me send you email. I'm in the middle of another class, so I'll send it by email. Because I don't even know what it is. I'm going to have to look it up. Okay, I'll send it to you by email. Oh, sure. Okay. Both phones ringing at the same time. A cell phone ring with spam from West Virginia, which I don't know what, and another student from a different class. Okay, so anyway. All right, so the first one, blind people exhibit the phenomenon of blind sight. So they duck if something if something goes toward their face, and they don't know why they even do. So, what's the nature of the blindness that these individuals have? Why are they blind? And why do they duck when they can't actually see anything? What's the problem?
The optic nerve.
No, actually, their optic nerve is working perfectly fine. Is there anything wrong with their eyes? Which usually is a person with a blindness? No. Otherwise, they wouldn't have ducked. So there's nothing wrong with their eyes or their optic nerve. So why are they blind?
The retina?
No, the retina is, like I said, the eyes are working perfectly well.
But if they're blind, they can't see. I don't understand.
Yeah. So if they can't see and yet they duck when something moves toward their head. Oh, wait a minute. Somebody's trying to get in. Hang on. Somebody's in the waiting room.
Okay.
All right. So the key is in the projection pathway for vision. And wait, let me show you what page that's on.
Good afternoon, sir.
Okay. On page 639 of the book. We're talking about the first question now. So you know what the projection pathway is. Where does the information go in the brain?
I'm sorry, what'd you ask?
Oh, the projection pathway. This is the key. The key to the answer is in the projection pathway for vision.
Isn't that your thymus?
Pathway means where does it go in the brain?
Isn't that the thymus?
What?
Thalamus.
Yeah, it goes to the thalamus. Yeah.
Okay.
Where else does it go? Well, where is vision usually processed?
Is the okay, never mind.
Where is vision?
The retina.
No, well, there's little processing in the retina, but I mean, really formation of the image and everything
Oh, okay.
occurs where?
Oh, at the primary visual cortex of the occipital lobe.
That's absolutely correct. That's where the vision is being processed. Hold on, so I guess another invite. Hang on. So it's going to the primary visual cortex. Remember, all the sensory information goes to a primary cortex. And remember, that's how your brain knows the difference between hearing, seeing, smelling, and where it goes in the brain, because your brain just sees action potentials, and every action potential looks like every other action potential. Okay, so it goes to primary visual cortex. So it looks like there's a problem in the primary visual cortex, like there's a lesion back there or something is disorganized, and that's why they're blind. With perfectly functioning eyes, they're blind because they have a brain problem, not an eye problem. Oh, wait. But where else does vision go? So, Lean, we just talked about, we were still in the first question. Talk about the answer to the first question. It lies in the projection pathway for vision. If you get your book, it's on page 639.
Thank you.
The occipital lobe?
It goes to occipital lobe, yeah. But that's for conscious vision. That's disturbed, so they can't, that's why they're blind. But it goes someplace else.
So is it the brainstem?
It doesn't, well, the top of the brainstem, what's that called?
Midbrain.
The what?
Midbrain.
It's the,
Medulla oblongata, or however you pronounce it.
no, it's not the medulla oblongata. That's in the middle,
The
it's
midbrain.
lower down in the brainstem. The top of the brain, I mean, the brainstem is the medulla oblongata, from going from inferior to superior, medulla oblongata, pons, midbrain.
Midbrain.
And so what is the structure in the midbrain where it goes?
The thalamus or the midbrain.
Well, it's right there on the right on page 639.
With the projectile nucleus of the midbrain.
And just below that,
Okay, superior calculus.
superior colliculus. If you remember, when we were studying the brain,
Right.
the corpora quadrigemini, there's four bodies. The two top one are superior colliculus and inferior colliculus. Inferior colliculus is auditory reflexes, and superior colliculus is visual reflexes. So that pathway is still there, and that part of the midbrain is perfectly good. There's nothing wrong with that ever. So this is like an unconscious vision, and not so that pathway is still there. And so when something comes at their face, that information goes to superior colliculus and makes them duck. Blind sight. And so what did this phenomenon suggest about the course of human evolution? Actually, I'm not, people are not writing down the answer to that. I'm still not marking it wrong. Because this is a really ancient pathway. Remember I talked about how the brain is constructed, the neocortex is all the 21st century bells and whistles, like in a brand new car, but down deep in the brain is a reptile and amphibian brain that's still there. It's like so you're making a 2021 car and you got to put it on a Model T chassis, the very first brains to work. And so this is a really amphibian pathway that's still in the brain. So it shows that we had an amphibian ancestor. Because a frog, if a frog is standing by the pond and you come up to it, it jumps in the pond. It sees you. But the only reason it saw you was because you moved. You stand there long enough and it comes out and it'll walk right over your shoes. If you don't move, it can't see you unless you move. If you remember that, and you remember that line from a very famous popular movie, didn't deal with amphibians, it's dealing with dinosaurs, Jurassic Park. This guy was making the assumption that dinosaurs saw in the same way. Remember, he was crouching there with one of the kids in front of the car, and the T-Rex is right in front of them, inches from their face. And what he tells the kid is, don't move. He can't see you if you don't move. So he was making an assumption that dinosaurs saw in the same way. That was a pretty dangerous assumption, because we don't know. Anyway, okay. So that's the answer to the first question. Second one, how is olfaction different from all the senses? And now it's again projection pathway. How is olfaction different? Now here it doesn't go someplace that all the senses go to.
Isn't it like smell is not responsive to chemical stimuli?
No, it is responsive to chemical stimuli. I mean, these are chemoreceptors, right? They respond to chemicals. That's what smell is.
It doesn't go through the thalamus.
It doesn't go to the thalamus. Every other sense goes to the thalamus. Where it goes is actually to, now we talk about the reptile brain now, and we talk about not the amphibian brain, but the reptile brain is the limbic system. It's the oldest part of your cerebrum. And so this is, and really smell is probably the oldest sense. And it's the most emotional sense, and the most sense that then bring back memories, because it goes right to the limbic system, where the amygdala is, that is the emotional center of the brain, and where the hippocampus is, where you consolidate, whoops, Roger is gone, where you consolidate memories. And so the olfactory goes right through that and then goes on to the cerebral cortex. It's also important to remember, though, that there is one pain pathway that bypasses the thalamus. One pain pathway, pain pathway bifurcates, and one, so pain does go through the thalamus, but one branch of pain goes to the limbic system, where emotional response to pain. Okay. And now, as far as regeneration is concerned, how are gustation and olfaction unique as far as regeneration is concerned?
You can regenerate your Like taste buds.
Yeah, your taste receptors, your olfactory receptors, they are regenerated, which is really, and they're neurons. So it's really strange because if you lose a hair cell, for example, in audition, it's gone. You lose, some retinal cells, they're gone. They don't come back. And then finally, a third part of that question is the auditory projection pathway. Here we go with another projection pathway. And let me go to that. Oh. So auditory projection pathway, 651. And I'll give you a hint, it's how many neurons are involved.
There's 4, right?
Yeah, there's one pathway is 4, one is 5. And how many are in all other sensory pathways?
3.
3. There's a primary, a secondary, and a tertiary. This one has a quaternary, a 4th one. So it's more, so that's how audition is unique. So I like these unique things about the senses. The senses are really strange, and there's a lot of unique stuff about them. And contradictory stuff. Well, on the next page, we'll talk about contradictory stuff that goes against what we've learned about the nervous system. Okay, so we got those.
So I had a question. What part of the olfactory gets regenerated?
Oh, the receptors themselves. It's a good point. I should say, how a gustation olfaction receptor is unique, because because the primary, secondary, and tertiary neurons, they don't get regenerated if they get destroyed, but the receptors themselves are replaced. Okay, so we're ready to fill in the blanks?
How are you? Good. All right. So this stuff is working well. Yeah, but this, because I also don't realize
Roger, you might want to mute.
I'm not muted.
So the first the first blank would be the senses of like the nose and the taste buds would be chemoreceptors or whatever, right?
Chemoreceptors,
Yeah,
correct.
chemo.
Those are the chemical senses, right? Okay, what about the general senses?
Are they also chemo?
No.
No.
It's the nosy, right?
No,
'Cause it's about the
no.
pain.
No, it's not the nose. What are the general senses?
Is it photo?
No.
Oh, man.
Well, first of all, what are the general senses?
Smell, taste.
No. So we need more basic information. What is the difference between the special senses and the general senses?
So is it general like touch?
Right. General means all over your body. All right. Special is around here, around your head. So vision a taste, a smell, vision, hearing is all special senses. But touch and pain and proprioception and pressure, that's all general,
General, okay.
which includes your head, but it's all of your body.
Okay.
So something touches you, those touch receptors, what kind of general type of receptors are they?
Mechanical.
Mechanoreceptors. They cause distortion of the membrane, like a Pacinian corpuscle in your skin. Push it, and you distort the membrane. It opens up ion channels. It stimulates a dendrite to threshold, and you get a action potential into your brain to the somatosensory cortex, and that part of your brain lights up. Because somebody just touched you on your right hand, on your right arm. Okay. Next, vision. This should be an easy one.
Photo.
Photoreceptors.
Mm-hmm.
Right. Then audition and equilibrium.
Mechano.
Those are mechanoreceptors also. Hair cells, remember, are used both in hearing and equilibrium, and they are mechanoreceptors. Again, distortion of the membrane. And then we go to proprioception.
Mechano.
And those are also mechano. So there's a lot of mechanoreceptors. But finally, pain, using process of elimination,
Is the noci or whatever?
those are the nociceptors.
There you go.
Okay, so we're done with the first page. So we're ready for the first question on the second page. Go back to vision. Describe how the membrane potential of photoreceptors changes in the dark and in the presence of light. And this is starting on page 636 and going to 638. Well, I'm going to add a little bit more to this question. How does this memory potential actually happen? Memory potential change actually happens. So what does happen to the memory potential of a photoreceptor in the dark versus the light?
Doesn't it have to do with something with like the rods and the cones in our
Well,
eye?
yes. So this is what we're talking about. We're talking about rods and cones. The memory potential of a rod or a cone.
Are they depolarized
When
in the dark?
they are depolarized in the dark, so about minus 40 millivolts. Why? It's because they have a lot of this stuff called cyclic GMP. Wait a minute, let me see now. Yeah, cyclic GMP. This stuff called cyclic GMP keeps open sodium ion channels in the dark. It's called the dark current. And so, so sodium comes in, rushes into the cell and keeps the photoreceptors depolarized. So what happens when you turn the light on?
The channel closes.
These channels close. That's correct. Now, this is not part of this question, but it is important for the exam to know why did they close? And that's on page 636, 637. Once the photon hits the photoreceptor, let's say a rod, it causes the bleaching reaction. You know what bleaching is? You have rhodopsin, the pigment in your eye, in rods. It's called scotopsin and cones, rhodopsin in rods. Let's look at rods. So it's a combination of opsin, a protein, and transretinal. Oh, wait. Cisretinal. Cisretinal was a vitamin A derivative. And so you get together. Once that photon hits, it causes the retinal to go from the cis formation that has a bend in it to a trans formation, which is straight. This detaches the retinal from the opsin. Okay, and then the opsin activates an enzyme that chews up cyclic GMP, and that closes the sodium ion channels and hyperpolarizes the rod. So it's bizarre, and the rods are depolarized in the dark. They're hyperpolarized in the light. And the next part of the question is, how does this change? How does hyperpolarizing a rod lead to an action potential in the ganglion cells? Because this is bizarre. Most receptors have to be turned on for you to get the sensation. They have to be depolarized, turned on. But vision is reversed. In order to see, you have to turn off your photoreceptors. You got to hyperpolarize them. Another incredibly bizarre thing in the senses. Okay, so remember, photoreceptors and bipolar. Now, photoreceptors are attached to bipolar cells. They have synapses with bipolar cells. Bipolar cells have synapses with ganglion cells. Ganglion cell axons form the optic nerve. So, so ganglion cells are the only cell here that we're talking about that connects an action potential. These other things are just gradient potentials. So, okay, so in the dark, the photoreceptor is depolarized, and so it's turned on, and it is dispensing inhibitory neurotransmitter onto the bipolar cells. So in the dark, it's inhibiting the bipolar cells. It's on, and it's turned off the bipolar cells. And the bipolar cells, since they're turned off, they turn on the ganglion cells. So the ganglion cells are off in the dark. See what I mean? Okay. You destroy the cyclic AMP, you close these sodium ion channels. The photoreceptor is turned off by being hyperpolarized, so it stops it. It stops those inhibitory synapses onto the bipolar cells are turned off. This activates the bipolar cell, which sends which turns on the synapses that it has with the ganglion cells, which are excitatory. Turns on the ganglion cell, brings the ganglion cells to threshold, and action potentials get shot into the optic nerve, into your brain. That's how it works.
Can
And
you
so-
repeat that one more time?
Okay, so in the dark, the photoreceptors are depolarized. They have inhibitory synapses on the bipolar cells. So when they're on, they're inhibiting the bipolar cells. They're sending out neurotransmitter and hyperpolarizing the bipolar cells, turning them off. So when the light comes on and the dark current is stopped and the photoreceptors are hyperpolarized, this turns off these inhibitory synapses on the bipolar cells. So now the bipolar cells are turned on. They're depolarized. They excite the ganglion cells because they have excitatory synapses at their base attached to ganglion cells. So ganglion cells are depolarized to threshold, and they send action potentials along the optic nerve, along their axons, which make up the optic nerve.
Thank
So
you.
how this goes against everything all other senses are activated is that in all other senses, you have to turn on the receptors to get the sensation. But in vision, you have to turn them off to get the sensation. Okay, so is everybody clear on that question?
Yes.
Okay. Then the last question, going to audition. So describe what happens to the membrane potential of a hair cell when a sound wave vibrates the basilar membrane. So remember, the basilar membrane is frequency coded. This is how we can tell pitch of sound by what part of the basilar membrane is vibrating. And what this does is it causes the hair cells in the organ of cortea are on the basilar membrane, and the stereocilia, remember, are stuck in the tectorial membrane, which doesn't move. So as you move the basilar membrane up and down, the stereocilia bend and they deform the membrane. And so how does the membrane potential of a hair cell change when its membrane is deformed? So we just talked about how all receptors work except vision when they're stimulated.
They're turned on.
Excuse me?
They're turned on then?
Yeah, they're turned on. So how do you turn on a nerve cell?
They hyperpolarize.
No, exact opposite. Hyperpolarizing
Yeah.
turns things off. Depolarizing turns things
Right.
on.
Okay.
So they are depolarized when their membrane is deformed. But what causes that depolarization?
Doesn't it have to do with potassium?
Right. So what does that have to do with potassium?
Doesn't it rush in?
Right. Potassium ion channels are open. Potassium rushes in. It's a positive charge ion, and that's what depolarizes the hair cell. And the last part of the question, why is that truly bizarre? It goes against everything you learned about the nervous system. Think of a regular nerve cell, how a regular nerve cell works. What happens to a regular nerve cell when you open up potassium ion channels?
Is it that usually potassium leaves?
Yes, potassium usually leaves cells. It doesn't come into cells. Because, and why does it leave? Because potassium is more concentrated inside nerve cells than it is outside nerve cells. And how does that change the membrane potential when that happens? Remember, potassium is a positive charged ion, and it's leaving the cell.
Can you repeat the question?
Okay, so in a regular nerve cell, when you open up potassium ion channels, potassium leaves cells. It doesn't come in, it leaves. And it's a positively charged ion. So how is it going to change the membrane potential of the cell in which it's leaving?
It hyperpolarization.
It causes hyperpolarization. It makes this inside cell more negative. That's the essence of an inhibitory synapse. Inhibitory synapses open up either chloride ion channels or potassium ion channels. And the falling phase of the action potential is due to opening up potassium ion channels. Okay. So why, but here in hair cells, the potassium is going in the opposite direction. It's going into cells and causing depolarization. Completely against, different from every other nerve cell in your body. And why is this? Well, what surrounds the hair cells? You hit this, it's a fluid. There's two fluids in your cochlea. One's paralymph in the scala tympani and scala vestibuli, and there's endolymph in the cochlear duct, which is where the hair cells are located. So what do we know about the composition of endolymph compared to the composition of the inside of the cell in terms of potassium?
Hyperpolarized. Hyperpolarized.
Yeah, it hyperpolarized, so it's going into cells. It's going into cells and depolarizing. So what do we know about endolymph? I mean, why are those potassium ions going into the hair cell? Why do ions cross membranes? I'm going back to square one here.
Doesn't the endolymph like help to allow sensory cells to function normally?
Well, yeah, yeah. But what is it about the endolymph that's so strange that causes potassium to enter a cell when potassium ion channels are open? Again, what's the process what's it called when when ions go across a membrane? What's that process called?
Like a concentration gradient or more?
Right, it's a concentration. This is simple diffusion. It's ions following a concentration gradient. So what do we know about endolymph then? As far as its potassium concentration goes.
Doesn't it have sodium and potassium?
Well,
Val?
yeah, but we're not worried about the sodium now, just the potassium.
So it has a high level of potassium.
It has a higher level of potassium than the intracellular fluid, which is completely bizarre, and because that's why the potassium goes in. In every other situation in your body, the nerve cells is lined with, nerve cells have interstitial fluid or cerebral spinal fluid, and their potassium and the potassium level in those fluids is really low. So and it's lower than the potassium level inside the cell. So if you open up potassium ion channels in those cells, potassium is going to leave. But endolymph has a really high potassium level, so high that it's higher than inside cells. And so in hair cells, when you open up a potassium ion channel, potassium follows its concentration gradient, goes into the cell and depolarizes it. So are we all set? Any other questions about this, about it, or about anything else?
Question. I was home, the letter that I sent you, so I didn't send this in yet.
Oh yeah. So that's the ANS activity.
Yeah. So should I upload it and send it to you?
Well, yeah, sure. Now, you drew in the pathways, the neurons in each one of those pathways.
Yes, sir.
Okay. All right. You can send it in to me if as some kind of attachment if you want. And you guys, if you want to, you can bring the group activity 5 to me either in a lab or to my office, or you can send me just a copy of it in an attachment, and I can download and look at it that way.
So just email it to you?
Yeah, I think you should be able to email it to me.
Okay.
Do we have an outline for the 5.5 exam?
Oh, no, as a matter of fact. Wait a
Okay.
minute, I may have, I mean, in the past, joints were part of the regular exam, so I might have a topics list about joints. I can look for one.
All right.
I could actually make one, but I think there's one already made in one of the from one of the other semesters. I'll take a look at it
Thank you, sir.
and see if I can put that up.
So I had a question just backtracking a little bit to the light and dark question.
Sure.
Okay, so most receptors need to be turned on, which is hyperpolarization.
No. Turning on in nerve cells, turning on is depolarization. Hyperpolarization is turning off.
So then the vision, they need to be depolarized to be turned on.
Well, yeah, and in the dark they're turned on. They're depolarized, so they're on in the dark.
Thanks.
Okay.
Is that everything or?
Yeah, that's well, that's all of group activity 5. So we're done with that. But if you, like I said, so we're done with the meeting. But if you got any other questions about anything, you might as well shoot them at me.
Oh yeah, so I did have, oh, sorry.
No, go ahead. I'm sorry. Go ahead.
So I did have a question. So the mechanoreceptors, what is that again? Like just general receptors?
Well, this is a receptor where deformation in the membrane opens up ion channels. So you deform the membrane, bend the membrane, and it
Yeah.
opens up ion channels. That's what a mechanoreceptor, how a mechanoreceptor works.
So is that like for like muscle movement or?
Oh, no, not really. We're going to get to the muscle chapter actually next. It's the last chapter, and they have to be. They're not receptors. I mean, their muscles work by stimulating an action potential in muscles to get them to contract. It's called excitation contraction coupling. So it's not. Now there are receptors, proprioceptors inside muscles that are wrapped around with nerves, and when those special muscle fibers move, they deform the membrane of the nerve that's wrapped around them, and this causes an action potential to go in to your cerebellum and tell you where your body parts are in space, how contracted your muscles are. That's proprioception. Those are mechanoreceptors as well. But the muscle is not a mechanoreceptor, but they have these muscle spindle fibers inside muscles that are mechanoreceptors.
So like touch is a mechano, right?
And touch is a mechanoreceptor,
Okay.
yeah.
Okay.
Thank you for your time and explaining this. I'm going to sign off.
Okay, that's good. Yeah, no, that was good.
Thank you, sir.
Okay, you're welcome.
All right.
All
Thank
right.
you.
Thank you. I'll see you next week.
Okay, we'll see you.
All right. Thank-
Bye-bye.